Thank you very much, Bill, for the kind introduction. And yeah, it's a pleasure to be here and And thanks also to ILSI IFBiC for the invitation to come and speak to you with you this morning. Is the volume okay now? You're okay? Okay. I guess just by way of introduction, I am recently appointed chief in the pre-market assessment division of the Bureau of Nutritional Sciences in Ottawa at Health Canada, and just wanted to let you know, especially my biotech company people here, that in my division my team's responsible for the assessment, mandatory pre-market assessment of infant formula, food additives, and health claims, and just to know that you're not alone in your concerns about the length of time it takes to review a submission, the number of questions we ask, the unpredictability, and all those concerns. So I get it from not just the biotech group. So this morning I wanted to talk about the nutritional aspects or the composition aspects of the food safety assessment of novel foods. Just by way of outline, I'd like to start off by just talking about the overall mandate of government agencies in the assessment of, or the assurance of safety of the food and feed supply. Talk a little bit about the Canadian situation, the role of Health Canada and CSIA, and then get into the elements of the safety assessment. What are the requirements in the submission of an application to have a safety assessment completed, how the compositional analysis fits into the overall assessment, what are the important elements to include, a little discussion about how much data we need, and then a little discussion at the end about plants that are modified for nutritional or health benefit. So Canada isn't any different from any other country that has a general oversight. All countries and national authorities have a role in the oversight of food and feed safety. And we heard a couple of times over the last few days that, you know, products of conventional breeding have no oversight whatsoever. They're not regulated. And that's a little bit of maybe a stretch, because we do have fundamentally each government agency has some sort of legislation in place that says thou shalt not sell an unsafe food. So there is a responsibility to, you know, in the basic legislation regarding food and feed safety. So in Canada, Health Canada has the responsibility for setting the standards and policies governing food safety and nutritional quality of food, including novel foods in Canada. Whereas the Canadian Food Inspection Agency, very closely working with Health Canada, is verifying the safety of livestock feeds that are manufactured and sold in Canada, or that are imported, to make sure those are safe, effective, and properly labeled. So, with globalization of the food supply and rapid advances in science and technology, that has resulted in a lot of foods being developed that have not previously been available in Canada. So in 1999, that's when the government of Canada introduced the novel foods regulations, and basically what those regulations say, they were an amendment to the Food and Drugs Act and regulations, and what those regulations say is that prior to the sale or advertising of a novel food, it must be notified to Health Canada and undergo a pre-market safety assessment. And in parallel, similar legislation, or regulations I should say, were brought in regarding novel feeds and also plants with novel traits. So what is a novel food? Here's where Canada differs slightly from other agencies. Different governmental departments and governments have decided on different methods of regulating plants. Plants and animals derived from modern biotechnology, but the definition in Canada is more broad. It basically, there are three basic types or situations in which the novel foods regulations would be triggered. So first of all, a novel food could be a substance that does not have a history of safe use as food in Canada. It could be a food that has been manufactured, prepared, processed, packaged in a way by using a process that hasn't been previously applied to the food and results in a major change to the food. An example of that might be high-pressure treatment of processed meats to reduce the microbiological or to improve the microbiological safety. That product would trigger the novel foods regulations and require that the product be notified to Health Canada and assessed prior to advertisement or sale. The third type is the one that we're most interested in this workshop about, and that is a food derived from a plant, animal, or microorganism that has been genetically modified in a way to either insert or add a characteristic that's not previously been observed in that plant, animal, or organism, or it no longer exhibits a trait, or one or more of the characteristics have been changed in a way that the product characteristics are outside of the range of what would be normally seen in that plant, animal, or microorganism. And so it's either the presence of a trait, the absence of a trait that's normally there, or a change to the range, outside of the range of normal variation for that substance. And the trigger, we heard from Laura yesterday, that it's an event-based system, so that each event is treated as a novel food. Just a word about pre-submission consultation. This is, before we get into all the principles and requirements, I wanted to just put out there that there's definitely, and we'll talk about it later, that there's definitely a case-by-case approach to novel foods assessments, and we really encourage pre-submission meetings to go over what is potentially proposed as the novel food coming in, first of all, to determine if it even triggers novelty and is required to be notified to Health Canada, to talk about what the product is, and to discuss what the data requirements would be. We find this really useful, and our feedback from petitioners is that it's really useful to either point out gaps where we think that it's very important to submit certain information, or maybe even, on the other hand, to discuss that certain requirements might not be needed. But so it could either go both ways, that we could have a clear expectation of what would be submitted and what would be expected in a submission. And we have recently published some guidance on our website about how to set up a pre-submission consultation. So the process. Generally, well, we do have guidelines for how the process works within Health Canada. Basically, the evaluation is conducted as a team approach with experts in all those areas coming together to work on the assessment. As Laura said yesterday, there are a lot of eyes looking at a submission. So this is just a kind of a process flow diagram, just to point out some of the steps that are done within our group. Basically, the novel food petitioner submits a submission to the novel food section at Health Canada, who are the coordinator of submissions. So they would send that submission out to the various bureaus, to the experts who are involved in that in the assessment. They would do their assessment, come together, put requirements, like either say yes, we're satisfied with the petition, or if there's additional questions, going back through the novel food section to iteratively go back and forth with the petition to work on those questions. Once we get to a point where all of the evaluators working on the submission are satisfied and ready to recommend that the food be approved, we prepare we call a food rulings proposal. So we basically have to go to our senior managers with a proposal to make a recommendation for approval or no objection of a novel food, and convince them that we've done our due diligence and we're ready to approve it. If that food rulings committee, which is a group of senior managers within the food directorate, it's kind of a intimidating procedure sometimes to go to them. They ask lots of questions, but if they're satisfied with our work, then a letter of no objection is sent to the petitioner. If they're not, they send us back to do more work, and we may be able to do that. Maybe simple clarifications, or we may need to go back to the petitioner with additional questions. So that's kind of the at the end of the process, the petitioner gets once approved, the petitioner gets a letter of no objection and a brief decision document published on the Health Canada website. So getting into the assessment principles, I wanted to talk to you about the basic principles that generally fall under Codex, and we've heard a lot over the last 2 days about the whole evolution of the process, starting with OECD, and I won't go into that, and then coming together of the ad hoc task force in Codex to come up with these principles and guidance. It's good to know that there previously it was kind of difficult to find all the elements of the Codex documents, but they've put them together into one edition that's either available on the web or hard copy like this that pulls together all the principles, the guidance, all the annexes about allergenicity, microorganisms, nutritional health benefits, so it's all easily accessible now. So to get into the assessment principles, our assessment principles, based largely in Codex. I guess the number one, or the one that I have listed up here is the tiering of data requirements is definitely the order of the day, that the degree of scientific support required can be adjusted based on the complexity and familiarity of the product, that it's very much a case-by-case, as I mentioned earlier, a case-by-case process, that because the products vary greatly, we don't have one set of prescribed data requirements, and they're determined on the basis of the characteristics of the product in question. We also are very tuned to the familiarity principle, that as we become more familiar with products and we become more familiar with assessing those products, that can be brought into the safety assessment as well. We will always accept a valid scientific rationale if a particular element that's listed in the guidance documents or in the Codex guidance documents is not in the petitioner's view required. We will always accept to review a scientific rationale for either not submitting data or bridging data. Also, a weight of evidence approach is used. It's basically the sum of all the overall data submitted, the assessment of that data that provides the context for determining the safety. Definitely that we're looking for the likelihood of unintended effects that may be present in the modified plan in question, and we're looking for the comparative approach, that the comparator must be appropriate for the product in question. For example, if you're, I guess, yeah, I'll have a slide on the comparators, maybe coming up in a moment. So for now, just say yes, appropriate comparator. In the Codex guidance, that is the closest non-GM relative. So general assessments of elements of the safety assessment, we've gone through this in a number of the talks already, that there are these aspects, broad aspects that all come together in the weight of evidence approach in the safety assessment, and that the nutritional and compositional assessment are one element of that safety assessment. I won't go into too much on the toxicology and allergenicity considerations other than to say that there are the two aspects. There's the endogenous toxins and allergens that may be present in the plant, the known toxin and allergens present in the plant, as well as the toxicity and allergenicity of the new material that are to be considered in the assessment. Chemical considerations may consider, and we would have our Bureau of Chem Safety involved in these aspects, to consider the potential that there could be elevated levels of chemical contaminants in food or feed, either through intended effects such as increased mineral absorption, those kind of traits, or those sorts of traits. So we would be looking, and also susceptibility to diseases or contaminants such as mycotoxins. So moving into the compositional assessment, we've heard this a couple times, but I think we'll repeat why we're doing a compositional assessment, or why we're requiring a compositional assessment as part of the safety assessment. That we are verifying that the genetic modification has not negatively impacted on the nutritional quality and safety of the food, and we are checking for unintended effects. So we're verifying that no unintended changes to nutritional composition or anti-nutrient composition have resulted from the modification, and verification that various nutrient and anti-nutrient levels in the plant are equivalent to a non-modified comparator and within the expected range. Some of the important elements, and we just heard a talk on one of the very important elements as well this morning, is to perform the compositional comparisons with an appropriate counterpart to determine the quality and consider the implications of any changes. For most plant species, and we have the OECD documents and other guidance to help us with this, but this is generally the list of the components that would be important to consider. That an appropriate comparator is used, and in Codex it's definitely the non-GM control, the isogenic or nearest parental relative. But other useful reference ranges that could be used are commercial varieties, of course, the ILSI crop composition database for the crops that it covers. GM varieties that have already been gone through the safety assessment could also provide information on ranges for nutrients. And in the case of nutritionally enhanced crops, even other foods may be appropriate to be used as comparators. What we struggle with, I guess, is how much data. Every time I get a new evaluator, that's when it really becomes, when a new staff member comes in to join the Bureau. They're scientists, they're curious, they want to know everything. So the list is long, and it's a constant discussion about what really needs to be submitted, what's nice to know versus what we need to know. And so just, I need you guys to feel my pain with that. So basically, and how many samples, how many reps, how many years, some of this we had a really good introduction with David's talk this morning. It's basically what we're saying with regard to data submission is we need a well-designed experiment. We need to know, we need it powered sufficiently and designed appropriately so that we'll pick up differences where they exist and where they're important. And you need to have a statistician, and we also rely heavily on the evaluation side on our statisticians within Health Canada. So it's a very important element at the beginning. So that's, I hope I used this statistically significant in the right context in this slide. I'm sure I'll hear about it if I didn't. So that's my question is, what do we do with all those statistically significant differences that we observe on the first run through the data package. And it goes back a bit to having the appropriate design in the first place to be able to detect the differences that you need. And the second bullet is kind of getting to what David introduced also, is what are we really looking for? Can we come up with ranges as suggested in the EFSA model that what is the biologically or the nutritionally important difference that would impact safety or nutritional quality? Is it 1%, 5, 10, 25%? Is it different for every nutrient? That's a discussion to have, and all that should be pre-planned. We can't go back, or we shouldn't go back to say, okay, that 1% difference is probably okay. We should know that before we go on our endeavor. I wanted to show, just getting to the statistical versus nutritional differences, as some examples of the kind of data that we've looked at, and we can see in this slide that this is a group of minerals. This is just selecting out the ones that were statistically significantly different in a whole range of minerals, and then just presenting on the first column the ILSI ranges in most cases. In some cases, there's other observations from OECD guidance documents when ILSI data is not available. And just to show the kind of numbers we see, the kind of numbers that come across our desk, where in each of these cases we would have said that yes, there's a statistical difference. Is it within the range? Are the means within the range, or is the range that we've observed within the range of the literature values? Are there any patterns when we look at different sites, and what's making up that mean? And basically coming to the conclusion that there are no significant, no nutritionally important differences in those for those minerals. We have seen some. This is another example where we do see an overall treatment effect in some cases. Again, I guess my point, yeah, for this one we're within the range of the literature values. There are some significant differences, and in this case, we've started to see some other statistical tests applied, such as the false discovery rate. And that's when I have to call my statistician, and they probably want to call the company's statistician to determine what's going on. But I guess the point is that these tools can be used with care, but it takes we have to make sure that we have the capacity to deal with that kind of data presentation, and we'll be coming to you to ask probably more questions. And we're not sure, and maybe David could comment when we get into the discussion about whether we needed to go, in this case, whether we needed to go to that step, whether we could have just done what we did in the previous slide and looked at the range, and whether this gives us any more assurance when we apply an additional test. This is a similar slide from another submission on minerals, and just to show the minerals again, this is just this reported significantly different report. Oh no, sorry, this is a kind of a blend slide, this third slide, where we have occurrence places where we see, like in line 2, where all the observed, all our experimental numbers are outside the range of the ILSI range, and yet there's a very to look at that and look at what the nutritional importance of that difference is helps us to make our decision that again is not a nutritionally or compositionally important difference. Again, this is another slide where we see that all the control, the sprayed, unsprayed, untreated, are all within the range of the literature values. There are some, they are statistically different. There's an application of a false discovery rate which narrows down or reduces the number of significant, statistically significant effects. And whether that's necessary, that's something that we're still discussing internally and discussing with petitioners, whether that's a necessary step. To get into potentially the novel foods improved for nutritional or health benefits, that was just kind of the place where we would see even more, I guess, more relevance of the compositional analysis, the importance of compositional analysis. There is some guidance on in Codex on the assessment and what are the differences in the assessment process for those improved crops versus the standard or the more familiar herbicide or insect tolerant input type traits. There's, the obvious, I guess, scope of that guidance is where a nutrient or another substance has been introduced to the plant, removed from the plant, an interaction between nutrients or other substances is happening. So for those types of novel foods, we would have the same basic assessment, and this came up in the roundtables yesterday in the group that I was in. That so far, I think that it has served us well to do the same basic assessment to isolate the intended change from potential unintended changes. Some of the additional considerations, though, for crops with enhanced nutrients or substances of health benefit are the bioavailability of the substance. Has that been impacted? And in some cases, well, we would, from a Health Canada perspective, where a crop was nutritionally enhanced, we would look at it from the whole diet perspective. We would want to know that we wouldn't, that this new crop would not negatively impact on the nutritional status of Canadians. We would use the nutrient databases. We would use our survey data to see what the impact of the introduction of that crop in a fairly conservative way to estimate what intakes could be. For example, I don't know, an increased selenium content of a crop, and whether that would put Canadians at risk of an overexposure to selenium, for example. As well, the appropriate comparator may be the non-GM control, but in addition, there could be the use of other foods that you're trying to, that are familiar in the food supply, that you would line up, say, a new fatty acid profile with a fatty acid profile, or a crop with a similar fatty acid profile to do that comparison. So just to wrap up, compositional assessment is an important element in the safety assessment of novel plants. Statistical differences should be investigated to determine relevance to nutrition and health, and the comparators and reference ranges are very useful in the interpretation and, of compositional analysis. A few websites that I'll include in the presentation for guidance, and thank you very much for your attention.
Good morning. I'm Owen Hookinger from the USDA. I had a question about the whole novel plant trait process. So I was wondering if the biofortified corn made to have high levels of provitamin A carotenoids that was created by combining rare alleles from different gene pools that are probably unlikely to be found in Canadian germplasm as at least one of them was tropical. But when you put them together using conventional breeding and marker-assisted selection, you can make something orange like a traffic cone that has highly bioavailable beta-carotene. So at what level of review would that sort of novel plant trait elicit if someone wanted to bring it into Canada for animal feed or other nutraceutical purposes? Thank you.
Thanks. Am I on? Okay. So with the question about the novel food approach in Canada and whether a conventionally bred, nutritionally enhanced product would trigger the novel food regulations, and the answer is probably yes. It's a subject of great discussion about how novel is novel, but so if there is a significant I shouldn't say significant, an important change to the nutritional composition of a food, then it would trigger the novel food regulations. The same with feed, and you mentioned nutraceuticals, and that's a separate agency, separate set of legislation, probably the Natural Health Products Regulation, and it would be notifiable as a supplement if it was going to be a supplement. Not necessarily because it's, just as all supplements are licensed in Canada.
Lynn, I was thinking about your, Russ Essner from Bear Crop Science, thank you. I was thinking about your slides on the adjusted p-values causing internal discussions within the agency. I was wondering what you think about petitioners coming forward with using the EFSA-style statistics for submissions to Canada. As a petitioner, I'm grappling with how to handle those the statistics coming out of EFSA on a global scale. I was wondering if you could comment on that.
That's a really good question. The question was about the use of the recently, I guess fairly recently published EFSA guidance on the equivalence ranges versus our traditional null hypothesis approach. And from my position, I think that's something that we would definitely entertain. It's something that we should discuss. We haven't had a lot of discussions. It's generally a case-by-case kind of approach as we receive petitions. So if I'm writing a note to make sure that we have a look at that internally within Health Canada so that we can be prepared. But it would be good to discuss that as a pre-submission kind of consultation before submitting the data for sure.